So I would like to introduce Ms. Norfilza Mohd Mokhtar from Universiti Kebangsaan Malaysia for the first talk. So time for you.
Okay, thank you, Ms. Chairman. I'll share. What about this?
Yes, now it's perfect.
All right, okay. So yeah, thank you so much, Ms. Chairman. So my topic today, first of all, I would like to thank the organizing committee to invite me. I'm going to talk on the title of the interaction of the gut microbiome and barrier function in the most common cancers in Malaysia, which is colorectal, and uprising cases is inflammatory bowel disease, but it is on the Malaysian perspective. So this is my outline of the talk. So I'm going to cover both on the gut microbiome and these two diseases and the role of intestinal barrier. And first of all, I would like to share the incidence of colorectal cancers based on the WHO of its Global Microbiome Report 2020, where it shows that 19.6% of our population per 100,000 population has colorectal cancer. And in terms of the ratio between male and female, it's about 1.3/1, which is a bit more common among male. So colorectal cancer is actually ranked the first among men and the second among women, and it is more common detected very late stage, stage 3 and stage 4, which is more than 70%, and more than 80% are detected at the age of more than 50, which is a bit late. Okay. So, but what we found from our setup is that in the last 2 decades, we actually looked into all the data, and we found that majority in our setup is in terms of the ethnicity, because you know that Malaysia has various ethnic, which is Malay, Chinese, and Indian. That's the main ethnicities in Malaysia, and the most common in our setup, in our medical center, is a Malay one as the colorectal cancer. Whereas not only that, male is a predominant compared to female. And what is more striking is when you see the cases from 2008 till 2017, the cases is going down, probably because a bit of the screening method is going on. But what is more rising is, of course, in 80 to 84 is still going up. But what is more striking is we see the trend between 40 to 49, that is increasing cases of colorectal cancers that is considered the onset is quite early, which is less than 50 years old. And this trend is more common among Malays, as you can see from this figure. So what is actually the reasons for the increasing trend among the younger age group? So we thought that we look at all the possible risk factor, which is actually the known cancer risk, cancer drivers, which is of course the obesity, alcohol consumption, cigarette smoking. Is there any difference between both? Family history is still the same, inflammatory bowel disease and diabetes mellitus. But after we have seen and we did publish on the review on this, and there is no exclusive risk factor for the early onset as compared to the late onset. So we thought that, okay, so is that answering the adenoma carcinoma hypothesis that's been published recently, where you know the from the adenoma till the carcinoma, there is an accumulation of mutation that is known, and of course the role of the inflammatory cytokine either in pro-inflammatory or anti-inflammatory, and of course this is not as simple as what you have seen in this figure. So what we thought that it could be the working hypothesis, it could be a connection between the the environmental triggers with the diet change in the diet of people in Malaysia, of course, that also with the underlying genetic defect that enhance the cause the increase in the permeability of the intestine, causing the bacterial product to be released into the circulation and causing a lot of problems. So we thought that that is the role of gut microbiota. So what we did is we actually come up with the design where we actually collected from the early onset polyp, which is less than 50, defined as less than 50, late onset polyp, and we also have a colorectal, young colorectal, which is also defined as less than 50, and also the usual more than 50 years old. And what we did is we collect the mucosal biopsy for the 16S sequencing, and we also did the dietary intake finding, and then using the FFQ, and of course we correlate that with the clinical findings. So what we found is that majority, as you can see here, of course, the majority are among the more than 50, which is very common. We can get it every week, and among male, and then Malay as well, and then the body mass index, majority are within their normal body mass index, which is not really a risk factor for this cancer, and of course there are no smoker. So what we did is the correlation of the 80 samples that we did the 16S sequencing, and we found that there is an association between a certain bacteria. For instance, in this case, Fusobacterium is significantly correlated with the vitamin D intake C and E, as well as others like Streptococcus agalactiae, which is actually strongly or significantly correlated with protein and cholesterol and so on. So there is a positive correlation and there is a negative correlation between the diet intake of all Malaysians, which is actually overall, we have a very low intake of fibers, which is actually around 5 grams, and the recommended intake is about 20 to 30 grams. And that is possible, all these correlations occur. So what we did is, of course, we sequenced the mucosal biopsy of this that we got from the 5 groups that I showed just now. So we saw that, of course, in the normal groups, the alpha diversity index is significantly different between all the groups as compared to the normal group, which has a very good diversity, and it getting less as the patient having either the polyp or the colorectal cancer. So therefore, we actually did the beta diversity, and we found it's significantly different between all the groups, and it's very well grouped all the patients according to the group that based on the clinical. So what we did is we actually look at the relative abundance based on the species, and we found that there is a pattern of the reduction of a good bacteria from the normal till the early onset, late onset, and of course the colorectal cancer as well. So what we actually come up with, we did a biomarker and network analysis, and we can actually come up with a specific biomarker that is actually age specific, which means that, as you can see here, there's a normal biomarker for a normal person, which is a Faecalibacterium, which is a known bacteria that is very good in a normal healthy people. And we also have people in the early onset polyp and early onset colorectal cancer. Early onset polyp is represented by the blue box, and it is actually seen in the Klebsiella as the biomarker for that. What about the red one? The red one is the early onset colorectal cancer, and it has its own group of gut microbiota that reflects the group. And of course, we have a late onset, which is the most, and the green one is the most, and we have a group of that. So in other words, what I can say that this biomarker could actually be validated in our Asian population or any other population that could actually reflect whether it can be used together with other, other relevant or the state of art of the biomarker that is available at the moment. So what we did is we also see the interaction from the So I just want to show it to you, the correlation or the interaction between the gut microbiota of all the groups that I mentioned just. You see the Faecalibacterium, the good one, and then and then it moved to the right one is the Fusobacterium. And these interactions are actually, we have to look into that more detail. And why is that? Some of the bacteria are very closely correlated with the network, and how is that it will lead to the pathogenesis of the disease. So these are the published data on the difference between the tumor-associated gut microbiota as well as the fecal microbiota that actually represent each area. So, so for instance, Shigella is reduced in the mucosal biopsy of a tumor associated that you get, and it increasing in the fecal. So, which means that it could be the biomarker. It depends on where does you actually profile the gut microbiota. Either it's from the fecal of the patient or from the mucosal biopsy of the tumor specific region. So this is another thing that already established in 2019, where it shows that Fusobacterium nucleatum is actually the known microbiota that actually represent, if it's elevated, it represent the advanced stage of colorectal cancer, as well as other type of bacteria, including the Ectodermium and Actinomyces, significantly increasing in multiple polypi. So another thing is that, as I mentioned just now, the risk factor of colorectal is we are looking at the inflammation-associated carcinogenesis. And in this case, in Malaysia, we are having increasing cases of IBD, inflammatory bowel disease. And what we did is we actually follow up the patient in prospective cohort. We follow up the patients in patients who are undergoing treatment, and then we did biopsy, and then we did take that for the electron microscope and so on. So what we found that, so we found that on the left side, the alpha diversity, unfortunately, because the post-treatment of the vedolizumab, which is anti-integrin for the moderate to severe cases of IDD, we didn't see any significant difference yet, because the pre-samples is quite huge number, but we couldn't get the post sample because we have to follow up the patient 3 months, 6 months, and so on. We still are collecting sample. This is just a preliminary. As you can see on the right side, it's still not significant yet because of the number of sample. I would say that the relative abundance at the phyla region has shown that there is significant changes, or no, not yet significant changes, but there is a pattern of reduction of good bacteria as in group of, for instance, firmicutes, and then it reduced after the post intervention. So the same goes at the species region. As you can see here, we take, for instance, Akkermansia muciniphila, which is actually a good bacteria. Is a gram-negative anaerobic. It is known to actually disrupt the mucin, but what happened is in the pre, it has got a lot, but in the post, it's reducing. It's supposed to be a beneficial. So we see that whether vedolizumab has some interference with the bacteria, the good bacteria, and kill the good bacteria. We're supposed to actually help in producing more it's actually inhibit it promotes anti-inflammatory cytokine, and it's actually inhibit pro-inflammatory cytokine. So we have to look into that in more detail. But what we did is we actually get the same sample, and we take the fecal slurries and we check the short chain fatty acid, which is actually supposed to be related with the dietary fibers. As I said initially, patients' dietary fibers is so much reduced for all patients, and we see that there is a difference between pre and post intervention. So we actually take the same.
You have 2 minutes.
Right. Okay.
Yeah.
Same patient, and we did electron microscope. We see the intestinal barrier, and this is an intestinal barrier of a normal patient, which has is tight junction, desmosome and gap junction, very tight to each other. That actually helps in the paracellular gaps and all that. So what happened is that when you have good response to the disease. As you can see, on the top is the non-responder, and the bottom is a good responder. And we see that the tight junction of the non-responder is still wider, and the tight junction of the responder is so much tight, and there's no significant difference between that and the normal one. But there is a significant difference between a non-responder to the responder as well as the normal one. So we confirm that with the expression data on the quantitative PCR, and we see that the expression of Claudin 8 is significantly elevated in the responder. And in the non-responder, even though there's no significant difference in other gene that is actually correspond to the tight junction, we saw that the occludin expression was significantly elevated in the non-responder. So overall, I would say that we actually, human is actually is as a result of the personal exposome. We are interact with the external exposome that gives rise to our biological response. And with that, I conclude that gut microbiome is still play an important role, which is actually cause the microorganism that actually can trigger the response that maintain the barrier function as well as tissue repair detected by the and is actually reflected by the genetic makeup. And this biosis, not only that, is actually reflected by, if you give any drugs, you can actually either make it worse or make it better in the whole thing. So we are still ongoing future research. We always focus. We will focus on the microbiome diversity and the outcome of the treatment. And with that, thank you for your attention.
Thank you very much for a very wonderful talk. And we will have a discussion as the last session.